Okay, good morning, everyone. Okay, just give me 1 second. All right. Yes, good morning, welcome, and thank you for joining us for this webinar on the molecular characterization of a sweet potato plant. I am Jean-Pierre Kavanaugh, and I am the visibility and PR consultant of the investment plan for the Caribbean regional track of the pilot program for climate resilience. Going forward, I'll refer to the project as a Caribbean Regional PPCR because the full project name is a mouthful. And I will be your host for this morning's webinar. Before we begin, I'd like to set some ground rules. The first is that I'm asking you not to unmute your mic while anyone is presenting. And if you want to make a comment or to ask a question, please use the raise hand function. How to raise your hand? There's a button at the bottom of your screen labeled raise hand. All you need to do is click it. A host or a co-host will unmute your mic and then you can start speaking. I'm asking you also to be aware of your video and audio settings to avoid static feedback. When your mic is muted, or when your mic is unmuted, by the host, please ensure that the device you're using is out of the direct path of the wind, because if, for example, the fan is blowing or the AC is blowing, then we'll hear it. And if you choose to turn on your question, please look directly into your camera. I'd like to especially welcome today's speakers and our main presenter. We have the Climate Change Senior Specialist at the Climate Change Division of the Inter-American Development Bank, Mr. Gerard P. Alleng. We have our Program Manager for the Caribbean Regional PPCR, Mr. Ainsley A. Henry. With the Country Representative of the Caribbean Agricultural Research and Development Institute, and going forward we'll refer to them as CARDI, Dr. Gregory. And our main presenter is the Senior Lecturer in Biochemistry at the University of the West Indies Cave Hill campus, Dr. Alleyne. Now, before we get into the presentations, I'll tell you a little bit more about our project. So the Caribbean Regional PPCR is a $10.39 million US dollar project designed to improve regional processes of climate relevant data capture, storage, analysis, access, transfer, and dissemination. And relevant to this webinar, to pilot and scale up innovative climate resilient initiatives. The project is executed through the Program Management Unit that falls under the University of the West Indies Mona Office of Research and Innovation. And if you're here us talking about the project, you may hear PMU Mori or PMU Mori. And there are 6 pilot countries. It's Dominica, Grenada, Haiti, Jamaica, St. Lucia, and St. Vincent and the Grenadines. Now, our program manager, that's Mr. Ainsley A. Henry, will give further insights about the Caribbean Regional PPCR. But before Ainsley speaks, I would like to welcome the Climate Change Senior Specialist from the IDB, Mr. Gerard Alleng, to bring greetings.
Thanks very much, Pierre, and I will follow your guidelines about looking into the video and making sure the fan is not on. Good morning, everyone. I hope everybody's doing well, especially given this new reality of working from home. For some of us who have been in lockdown for the past 4 months, it's a interesting experience. But certainly, I'm sure everybody is adjusting and being able to function, and please continue doing so. Really on behalf of the Inter-American Development Bank, we're really pleased to support this study on the molecular characterization of sweet potato varieties within the Caribbean, as part, as Jean-Pierre indicated, of the Caribbean regional track of the pilot program for climate resilience, or PPCR. Some of you may know that the regional track of the program started about 5 years ago with the objective of helping countries within the region adapt to climate change through various regional and country interventions, including improvements to climate relevant data acquisition, storage, analysis, access, transfer, and dissemination of information. and to pilot and scale up innovative climate resilient initiatives. It's under this component of innovative or adaptive initiatives that this study resides. You know, we all recognize that climate change represents a major risk to food security, to crop yields, and to the broader economic performance of the agricultural sector. And I hope I'm speaking to the choir on that. And two of the more significant climate risk factors to the sector in the region include reduced water availability and extreme rainfall events. Although increased heat, pests and pathogens are also ranked really high. Trying to maintain crop production following a conventional approach to crop management and to plant breeding may no longer be sustainable or will be challenging, given that we will be living, already sort of living in a warmer world. Whether we maintain average temperatures at 1.5 degrees Celsius, as CARICOM continues to push hard on with the Paris Agreement, or even at a higher level. Advances in genomic technologies, capacity, and knowledge will provide us with important tools to help the agricultural sector adapt to a rapidly changing climate, especially to help reduce the time to produce new varieties that are also climate ready for the new climate regime. For this reason, I think this molecular characterization study is important, and it advances our understanding of the varieties of a crop that have drought or soil tolerant properties. We definitely will have to do more of this type of investigation, as there's a range of economically important crops in the region that we need to characterize in terms of climate resilience. Future food production in the Caribbean will rely on the development of new crop varieties. And that starts with a better understanding of what is available, what has resilient characteristics, and how they can be developed in a faster time period. So for me and for the bank, we really look forward to the presentation of the results of the study and the implications with regard to improving resilience within the agricultural sector. Thank you very much.
Okay, thank you so much, Gerard. All right, so we'll move in. We'll now ask the program manager of the Caribbean Regional PPCR, Mr. Ainsley Henry, to say a few words.
Good morning, everyone. Thank you, Jean-Pierre. First, let me start by welcoming you on behalf of the investment plan for the Caribbean Regional Track of the Pilot Program for Climate Resilience, as Jean-Pierre mentioned, a very long name, but this is the name of the project. Additionally, the University of the West Indies Mona campus and the Mona Office of Research and Innovation, which is where we're housed. Now, the scope of activities under this project is really quite large. It ranges from health, fisheries, meteorology, geospatial information and climate modeling to the topic of today's activity, which is agriculture. The Caribbean Agricultural Research and Development Institute is a key partner in the implementation of all agriculture-related activities under this project. And together, we have been involved in a wide range of activities related to agriculture. These include building infrastructure, such as a seed bank, gene bank, weaning and hardening facilities, as well as increasing the human capacity of the agriculturalists in the region through a variety of training opportunities. To bring context to today's discussion, it's interesting to note that hundreds of years before the advent of Christ and conquistadors, sweet potatoes being cultivated in Central and South America is an important food source. In modern times, it's been a major cash crop across the world, using approximately 7,000 varieties. As Gerard mentioned, with this wide scope of varieties, it means that there's a large possibility, a large opportunity to build a better plant through selective breeding. And the key to doing that is understanding exactly what opportunities exist in the plant through work such as that which has been done today. Now, we here in the Caribbean have long recognized the importance of the crop for its local dietary and economic benefits, and hence it's been a staple for farmers for many years. In the context of a changing climate and the increasing importance of food security, the diversity of this crop, and in particular, its resilience in the face of droughts, high temperatures, and increased need for salt tolerance, makes it an excellent crop for our countries. It is with this in mind that we have contracted one of our excellent regional scientists, Dr. Alleyne, to do a molecular characterization of these properties and more of this important crop. I am certain that the information presented today will form another tool in our arsenal to build climate resilience across the region. And again, I welcome you and I wish you a productive session. Thank you.
Okay, thank you. Ainsley, as Ainsley had mentioned, our project's execution in agriculture would not be possible without our partnership with CARDI. Now, Dr. Gregory from CARDI will now speak. He's a country representative for Jamaica, by the way. And he will now speak about why sweet potato was selected, along with some other interesting information about the plant. Gregory?
My screen?
Yes, I've seen your screen, Greg.
All right, okay. Good morning, all. First, let me recognize my colleagues from CARDI, my colleagues from AICA, FAO, the Ministries of Agriculture from across the region who have worked with us on this project. My presentation is really a prelude to the main presentation, molecular characterization of sweet potato. And I will briefly talk about the importance of sweet potato to the Caribbean region and CARDI's role in developing the sweet potato industry in the region, and how we, over the years, have ended up with a very interesting molecular study funded by the project, the PPCR project. The outline of my presentation is as follows. I will talk about sweet potato as a superfood. I'll give an overview of the sweet potato in the Caribbean, CARDI's role in developing the sweet potato industry, and CARDI's future research for development. Sweet potato, as mentioned by the program manager a while ago, is a superfood. It's a crop of growing importance to the region. It's widely recognized as a potential health and wellness commodity. It's a good substitute for the simple carbohydrates, which are among the main causes of chronic non-communicable diseases. It's rich in beta-carotene, minerals like calcium and iron, antioxidants, and fiber. If we look at the nutritional profile of sweet potato and potato, where they are not comparable, sweet potato is superior in fiber, calcium, iron, vitamin A, and vitamin E, just making it not only a staple, but a very nutritious staple for consumers in the Caribbean. All the CARICOM countries grow sweet potato, but by far the largest producers and exporters of sweet potato in CARICOM are Jamaica and St. Vincent and the Grenadines. Sweet potato is mainly grown by small farmers. It's highly adaptable to a range of growing conditions. That is how we viewed sweet potato years ago, highly adaptable. However, we now realize it has drought-tolerant properties. It's also very low, it has a low susceptibility to natural disasters, and therefore it has potential as a climate change adaptation crop. And that was highly recognized in the passage, after the passage of Hurricanes Marie and Dorian recently. Productivity is below the agronomic potential of the crop. The region's average production yield is 7,900 kilograms per hectare. The world average is 11,400 kilograms per hectare. Jamaica's average yield, however, is 18,000 100 kilograms per hectare, above the international average. There's some challenges to the production of sweet potato, and mainly the incidence of pests, the weevil and the grub are the ones that are the most constraining to production. Sweet potato has a good shelf life. Value-added products ranging from flour and paper to wood and pharmaceuticals offer market and investment potential. In 2019, the region imported 60,000 tons of frozen potatoes, US value $57.4 million. That gives the producers in the region good opportunities for import substitution. I'll briefly talk now about the role of CARDIs in the development of the sweet potato industry in the region. As far back as 2000, CARICOM gave CARDI a mandate to develop the sweet potato industry in the Caribbean. Since then, our work has focused on strengthening the value chain, and the priority areas were increasing productivity, value-added product development, morphological characterization, biochemical analysis, molecular characterization, and drought-tolerant studies. While looking at increasing productivity, we introduced a number of high-yielding cultivars from SIP. We conducted agronomy trials across the region. We conducted integrated pest management studies, which promoted the use of pheromone traps and cultural practices as a way of controlling the disease. And we developed practices for controlling and managing the grub, which is another soil-bound insect that scars the tuber. It is clear to us when we did post-harvest handling studies that curing prolongs the shelf life of sweet potato. It's a very simple technique. Once you can ensure that the facility has a temperature ranging between 26 and 29 degrees centigrade, a relative humidity of 85 to 90%, and you can cure for 4 to 7 days. Results from our agronomy trials have shown that varieties, the time of planting, and the agroecological zones impact on sweet potato yield. And therefore, production planning will have to take those into consideration when we're developing marketing and production systems for export, food and nutritional security, and local consumption. Value-added product development is an area we spent a lot of time, and we support from the CARICOM Japan project, the APP EU-funded project, and in collaboration with FAO, we've identified what the most common varieties used in the region are best for. And there's a list here which shows what is best for flour, paste, juice, and fries. These are some of the products, value-added products that we can make from sweet potato. From composite bread, which is a combination of grated fresh sweet potato, 40%, 60% wheat flour, and that was introduced to bakers across the region. The commodity, the product, high price, well sorted out, well in high demand. However, the cost of the raw material made it prohibitive for the bakers to have any commercial ventures with that commodity. Morphological characterization. This is the way we use to differentiate sweet potato varieties in the region. That's the traditional way we use, which is a photo documentation technique. We normally would have germplasm banks, and where there are no tissue culture labs in the country, these banks serve as conservation and multiplication plots for small farmers. Morphological characterization is part of a process of standardizing genotypes in the region. And we will see some of that in the next presentation. Drought tolerance studies are being conducted. Last year, we did a drought tolerance study on the CARDI station, and clearly it showed that ganja and uplifter are two of the varieties that have drought tolerant tendencies. CARDI's future areas for research. We want to continue molecular technique studies. We want to do drought tolerance studies. We want to expand the production systems to produce virus-free planting material for farmers. We need to do technology transfer using ICT tools, because in the advent of COVID, we realize we have to change our focus and methods of reaching the farmer. We need to continue organoleptic and nutritional analysis studies, determine varieties best suited for specific value added products. Production planning is important, as I mentioned before, and scheduling to produce higher volumes at minimal cost. We also developing investment profiles for value added products like fries, crisps, and flour. And these profiles will support small businesses when they seek funding. The top 3 bullets I have highlighted, and I will start with bullet 3, where the CARDI's plan in the future to expand the production system for production of virus-free planting material. As I speak, the PPCR project has provided funding for greenhouses to be established for planting material, clean planting material development in Jamaica, Dominica, St. Lucia, and Grenada. The facility in Jamaica has been erected. It has been outfitted with an irrigation system, which will be retrofitted to produce clean planting material of sweet potato in pots. These greenhouses have screens that are vector-proof and will prevent the transfer of any diseases through pests. Drought tolerance studies, the project, the PPCR project, again has provided resources under the agricultural component to support studies which are ongoing in Jamaica on sweet potato, and we're actually repeating the experiment done last year to ensure that we have the scientific integrity in the results that will go also to support the modeling and the use of the DSAT and AquaCrop models. And I should remind CARDI staff and the region that the project did train our young scientists in a basic and advanced course in crop modeling. So the validation plots, which are drought tolerance studies, will support that venture. Now we come to the focal point of today, molecular characterization. We've just completed a study in 5 pilot countries. Again, that study is supported by PPCR funding. And the study was conducted in Dominica, St. Lucia, St. Vincent, Grenada, and Jamaica. Because Dr. Alleyne is based in Barbados, Cable Campus, where she conducted all the analysis, we incorporated a couple accessions from Barbados in this study. That study has been completed and is going to be the subject of the next presentation. It is interesting to find out and to see how the drought tolerance studies done in Jamaica on sweet potato compare with the actual findings from the molecular studies at the genetic level. And if they're comparable, it would be a real novel experience and deliverable from the project. Before I close, I would like to thank the project management that has provided really good support to CARDI in the form of Ainsley Henry, the program manager, Erica Houghton, who's behind the scenes as the procurement specialist, and the newly appointed technical coordinator who replaced Rashida Hall Hansen, there's Arena Delancey. Thank you, and back to the moderator.
All right, thank you so much, Dr. Robin. CARDI's work in sweet potato remains relevant to the region, and I'm hoping that we'll be able to move forward with even more research into the sweet potato plant, along with moving this research into the real world. So planting with climate in mind, as well as filling the gaps that exist and doing more value-added products. Now, before Dr. Alleyne speaks, a little bit on her background. Dr. Alleyne is a biochemist and biologist at the University of the West Indies Cave Hill campus, Barbados. She earned her PhD in biology studying yam anthracnose disease caused in Barbados. She also spent 2 years as a postdoctoral research fellow at the University of Nebraska in Lincoln, Nebraska, USA. She holds postdoctoral certificates in diagnostics at the Michigan State University and the University of South Carolina. She specializes in plant pathology and is a coordinator of the MSc in Biosafety degree program at the UWI Cave Hill. She's a highly published academic with more than 25 peer-published journal articles and published conference proceedings, along with several book chapters and technical reports. She's also senior editor of Plant Health Progress and American Physical Society Journal. Her research projects are varied and currently include studies of diseases in fruit crops, cassava, sweet potato, and yam in Barbados. Now, Dr. Alleyne, on to your presentation.
Good morning, all, and good morning, Jean-Pierre. Thank you for that introduction. And it's a pleasure to be here and follow on from Dr. Greg Robin's introduction onto the sweet potato. Without further ado, let me just share my screen and get us going. Okay. Okay. I hope you can all see the screen.
Yes, we're seeing the screen.
Thank you. And so this morning, as our presenters have all said, we have been working on a small aspect of the Caribbean PPCR project, the molecular characterization of sweet potato. Before I get into the presentation, I just want to tell you a bit about the PPCR and what the aim of that project is and how our little project fell underneath that big umbrella of the PPCR. I'm sure you would have heard previously from other speakers that the Caribbean PPCR, its main goal is to improve data acquisition processes in relation to climate resilience. And our project falls under the component of agriculture. And specifically, we used molecular techniques to identify the varieties of sweet potatoes in 6 of the islands that have shown some drought tolerance. So in our presentation this morning, I am going to give you a background on the sweet potato. I will give you a brief introduction to genome analysis, or one aspect of genome analysis. We will spend some time talking about how the varieties were sampled, and then what we did to investigate the genome of these different sweet potatoes that we had. I will present the results of that investigation and discuss those results and end with a few brief concluding remarks. So some of you may or may not know that sweet potato belongs to a large family called Convolvulaceae, and one of its most prominent members is the morning glory. We've all seen these flowers in the wild in our region, growing on the roadsides and so forth. Well, this is sweet potato, and as you can see, it resembles the morning glory quite well. So it's part of that large family of plants. With this large family of plants, and within sweet potato itself, we have a wide range of variation in the genetics of the plant. We have variation in terms of its leaf shape. We have triangular, lobular shapes. We have even highly lobed varieties in terms of the shape. And we have semi-lobed varieties. So we can have that kind of variation in our plant. We have variation in terms of skin color. We have white skin types. We have pink skin types. And we even have reddish orange skin types. And you would have heard Dr. Robin indicate that this method of just visually looking at the plant based on its skin and also its flesh color were traditional ways in which we described our sweet potato varieties. We have in some of the islands purple skin, purple flesh colors. We have examples of white skin color with pink skin, white flesh with pink skin. We even have deep orange flesh colors with pink skin and pink and yellow or cream skin varieties. So it's a wide range. Interestingly, as Dr. Robin said earlier, traditionally we've had these varieties and named them based on what they've looked like. And so you have some interesting names within the region. So for example, one that comes out at me is something called Fire on Land from Jamaica. I'm not sure what they were thinking about, but that's what they described it as. We have varieties like Yellow Belly from Jamaica. We have varieties from St. Vincent called William Blue Top. So there is a lot of varieties and a lot of local names given to these varieties. However, these varieties, in terms of what they are, may be the same variety in one island, but we just don't know because the name is local, and it may also not match up to the name in the specific gene banks housed at the International Potato Center, or SIP in Peru, which has over 5,500 accessions. So there's a lot of sweet potato types globally. This is the largest repository, the SIP Center in Peru. The US Germplasm Bank has more than 700 accessions, and they've recently looked at the characterization of their germplasm. It's not completed yet, and there's still ongoing work on that. They're again using tools such as markers, molecular markers to do so. And I want you to bear in mind that in our study, we've begun in the region to describe our genome through next generation sequencing tools. So not using traditional molecular markers, but genome sequencing. In our region, based on CARDI's data, we have about approximately 120 accessions of sweet potato, and this excludes Guyana, and I know Guyana is beginning to look at their varieties as well. So we will have a lot more in our data bank. But the interesting thing about sweet potato and having so many different varieties with so many different names of so many different colors and variation in terms of its morphology is that it maintains a broad gene pool for us. And that broad gene pool, as you would have heard by others this morning, provides us with an opportunity in the advent of climate change to begin to interrogate that genome, to pull out those genes or to look at those genes that will assist us in building the type of sweet potato for climate smart agriculture in the future. This gene pool is basically the bedrock of our biodiversity, and it allows us to have genes that will look at things like pest and disease resistance. The topic of today's talk, drought tolerance. We will be able to look at genes that will help us increase yield and also improve our nutrient characteristics. So there's a lot of opportunity when you look at the genome and the biodiversity that we have in the region. The sweet potato genome is a very complex one. It is described as hexaploid and highly heterozygous. What does that mean? It simply means we have multiple copies of the chromosome number. As human beings, we all have 23 pairs of 46 chromosomes. Sweet potato has 90 chromosomes. Many of those chromosomes have been repeated over and over and over again, And that makes mixing of the genetic material very common, and hence that's why it's heterozygous. Because it's such a complex genome to study, there have been only 2 projects that have looked at the complete genome. And I say complete because that's how they published it, but these genomes are still incomplete. One of the main projects is the GT4SP project, housed at the University of Michigan, funded by the Bill and Melinda Gates Foundation. And the next one is the NCBI genome IPO-BAT genome. And this is a genome that is housed at the NCBI Gene Bank Repository in Washington, DC. And both of these are publicly available, the assemblies. An assembly in a genome simply means you have read all the DNA inside of that genome. You've read all the genes on the chromosomes, or you've tried to, and then you've reassembled that, you've joined them back together and created a database so that you can study and match and see what's in there. In the GT4SP project, they have looked at 2 wild types, Ipomea trifida and Ipomea triloba. These are the closer relatives of wild sweet potato. And in the NCBI IPO-BAT study, they haven't used a Chinese variety called Taizong, and this has been housed in the GenBank study. However, the results of those studies indicated that while Ipomea batatas has been sequenced, it has not been fully sequenced, while the closest relative, wild relative, Ipomea trifida, has been fully sequenced as much as possible and has a better sequence information in the databases than Ipomea batatas, this Chinese variety here. And so based on that information, the Ipomea trifida was the reference strain for us in terms of matching to the varieties we have in the region. So coming back to our methodology now, we simply got leaf samples collected from different varieties across the 6 countries when we included Barbados. We extracted DNA in my lab. We did DNA quantification. In other words, we made sure the DNA was of good quality. And then we sent that to a commercial entity for gene sequencing. Gene sequencing or genome sequencing at this level is done by several high throughput machines and cannot really be done in one particular lab. And so it's a commercial enterprise, but that information is sent back to us, and then we begin to use that information to do the data analysis. So again, here are the steps we got from each country. They sent us approximately 15 leaves, 3 leaves per variety, and there were 5 varieties from each country. These leaves were sent as a dried sample, and the DNA is still intact in that sense. It's sent that way to make sure we're not transmitting any diseases and so forth. And they were shipped to the lab. And in the lab, this is how the leaves came. We then extracted DNA and quantified that DNA. Once the DNA was quantified and purified by fluorescence, it was sent to the sequencing facility where a genomic library is made. A library basically is a set of sequences that you can then pull out the specific references that you want. This is done, and then it is sequenced using next generation techniques. Once the data has come back to us, then we build what is called phylogenetic trees or family trees and compare that to our reference genome. And bearing in mind, our reference is still Ipomea trifida. So again, to describe that in 3 short steps, you have your genomic DNA, and the sequencing produces what is called paired end reads, which are short bits of sequence, and then that is aligned, and many copies here because it's a whole genome, it's aligned to our reference. So from those samples that we had, 20 samples were taken from the islands and sent for next-gen sequencing, simply because when we looked at the DNA quality, the purity, and the cost, we were able to send 20 samples. We had 3 from Grenada, and we had Hockey, cream flesh, pale pink skin. Cricket Gill, these are local names now. Bright red skin and white or cream flesh. Reggie, a pink skin with white flesh. And you can see the variation in leaf forms underneath here. From Jamaica, we had 4 accessions. We had Ganja, a lobe variety with pink skin and cream flesh. We had Uplifter, a pink or red skin with a very bright yellow flesh. We had Yellow Belly, again, somewhat circular in shape with pink skin and yellow flesh, pale yellow flesh. We had Fire on Land, and again, it's a lobe variety with pale pink skin and cream flesh. From Dominica, we had no local names. However, we gave those descriptors based on their morphologies. And so we had a deep purple skin and white flesh variety from Dominica. We named that SDPW01. We had a purple skin, yellow flesh variety, SDRY01. A purple skin, purple flesh variety. You can see here, nice purple pickling on the inside. It was called SDPP01. And then we had a cream skin, white flesh, one of those from Dominica, SDCW01. From St. Lucia, we had 4. Most of the St. Lucia types were white to pale white cream skin and white to pale yellow to orange skin varieties. Again, there were no local names available for the ones from St. Lucia, and we had 1 red skin, white flesh. You can see it's summer disease, but it's a red skin, white flesh variety. From St. Vincent, we had 4, Agriculture, Black Vine, CARDI, and I can't recall the name of this one, but they were all quite similar, smooth, red or pink skin with yellow flesh. We also, because I work in Barbados, included one of the more popular varieties from Barbados, C104, which is a red skin, cream flesh variety, or pink skin. Once we got the data back, the data was visualized. Again, we're talking about lots of genomic sequences, and these are the 4 letters we get from the nucleotide sequences. And so you have to look at that visually. And so we use the GT4SP project, which has a genome browser, and we interrogated it against the reference sequence, iTriFeeder, using their browser here, JBrowse. We also use another program called SNPplay, which is used to look for variations in large data sets. So coming out of that, we discovered, and this was not an unexpected result, that the genome is highly heterozygous, some very, lots of sequences, some very dense areas of genetic sequences, and some very sparse areas. This would come because of the hexaploid nature or polyploid nature of the genome and the genetic mixing that occurs. We also found that there were more transitions than transversions. So for those geneticists among us, a transition simply means when you are moving or having a mutation in the genome or in the genetic sequence of one base pair into another. And a base pair simply means those 4 letters that we know so well: adenine, cytosine, guanine, A, G, T, and C, when those can be changed from one into another. So if you're changing the same base, it's called a transition. If you're changing 2 different types of bases, it's called a transversion. Of course, transversions require much more energy than transitions. And as you can see, for most genomes, therefore, transitions are quite normal, and they tend to have more of those than transversions. This is what we saw in our genome. So we were quite pleased that up to this point, the data was fairly robust. When we looked at the data in the genome browser from the GT4SP project, we saw that we got some hits in the genome for our red or pink skin and white and cream flesh varieties. In other words, all of varieties on this gene called IBMBY1, And in the gene bank, it has a very long title here. So we downloaded that sequence, and then we use that information to look at the relationships among our varieties. That gene actually codes for anthocyanins. And anthocyanins are these polyphenols, these large organic molecules that you can find in plants that give them the range of the different colors that they have. Interestingly enough, anthocyanins can protect plants from abiotic stresses. We're talking things like high temperatures and so on. In sweet potato, the purple ones for sure and the red ones, the anthocyanins are called cyanidins and pianidins, and it simply means they have a substitution in the molecular structure in this polyphenol here. In sweet potato, most of our red and pink skin varieties and white and yellow flesh varieties, they have this anthocyanin production. So the gene is there in all the varieties that we saw. It is expressed fully in the tuber flesh and not much in the other parts of the plant, however, in sweet potato. And it is known from a different study that there is more of the anthocyanin in some deep red varieties than the yellow flesh or white skin varieties. So using that information, we got all the genes and a reference gene for IBMYB1 from GenBank in the database. And we began to see in our varieties how they matched up. And we used a technique called RAX-ML, which is simply put a way of drawing a family tree using phylogeny. And it gives us maximum likelihood substitutions probability. And we got this tree. And the take-home message from this simply is that when we looked at all the varieties, they all had this anthocyanin gene, but we found that they tended to group somewhat by location. So here we have most of St. Lucia types. Here we have most of our Grenada types. Here we have, again, Jamaica types here. But we did have some differences. So we have, for example, here, a white flesh and a pink skin with a white skin and white or yellow flesh variety in this clade here. This is our gene. So it just shows that they all have the gene and they seem to be clustering in this particular analysis by location. This study did not include the St. Vincent nor the Barbados isolations, simply because in our next study we found that those were absent, and so we made both studies the same in terms of what we were looking for at this point in time. When we looked at drought tolerance, we also got hits in the database from the genome browser. Again, so we downloaded that gene that we found, which was the abscisic acid binding responsive element, or ABRE factor, and it's abbreviated IBABF4. IB meaning Ipomoea batatas, abscisic acid binding responsive element 4, IBABF4. But we found that when we looked at the Barbados variety, the one we had, there were no mutations or genes that were mutated for that in the C104. We could not find any hits in our database for C104 and this particular drought resistance gene. The same thing happened with the isolates from St. Vincent. We could not find any hits in the database for those, for this particular drought resistance gene in the St. Vincent isolations. We found it in all the others, and we found that some St. Lucia clustered together, etc. So when we looked at this gene further, it is what is called a transcription factor. It simply means that in making of proteins, it is involved in the making of proteins, and in the making of proteins that assist the plant in being tolerant to drought, salt, and oxidative stress. This means that this binding factor here binds abscisic acid, which is a hormone that increases the plant's ability to withstand drought, high salt, And in doing so, it leads to closing of the stomata in the plant and may lead to better photosynthetic efficiency or greater efficiency during photosynthesis. Of note, the only yellow flesh variety that had this hit was the uplifter from Jamaica. The black vine, agriculture, and St. Vincent types did not have this gene, and so they were not included in our analysis for looking at the similarity, nor did the C104 from Barbados. When we did our phylogenetics or our family tree analysis, we've got 3 large groups. Again, it was done using this maximum likelihood method. It gives us probability statistics for large genome data sets. And we found in the group that had the ABF4 gene, we found ganja, this SDPW from Dominica, crooked gill from Grenada, this unnamed pink white flesh variety from Dominica, SDRY, and we also found one of the yellow fleshed varieties from St. Lucia. The next closest group to this large group here with our gene was number 2, a smaller group, but it also included the pale pink skins and white flesh. There was one outlier here with this white skin, yellow flesh, but pale yellow flesh from St. Lucia. But again, here we had hockey from Grenada and fire on land from Jamaica. The rest of the isolates did have the gene, but they all clustered together in a very large group that could not be teased out. There were 2 subgroups, for example, one where we had the paler varieties and one where we had a mixture of yellow, purple, and so on. So the take-home note here is that our pale pink skin and red skin varieties seem to be giving us, have a good relationship with this ABF4 gene for drought tolerance and salt tolerance. We began to look further at the interactions of that gene with other genes in the genome. And so when we did a network analysis, we took that gene out and looked to see in the functional analysis, what does that gene do exactly? We found it had a lot of interactions with other drought resistance genes. So for example, ABF4 can interact with a calcium dependent protein. It can also interact with something called a dehydration responsive binding protein. Again, binding proteins involved in drought tolerance. And it also interacts with other ABF genes, abscisic binding proteins. Now, abscisic acid is a known protein involved in drought tolerance. And so in summary, with other drought tolerance genes, we have one outlier from St. Lucia here, and this can be looked at further. We also know that the yellow types the yellow flesh varieties do not have these genes. So the take-home message is that drought-resistant and tolerant field trials should therefore be conducted for our red skin, white flesh types in the region. And I am pleased to note that this has begun in Jamaica. And it's interesting that I saw in the early presentation that ganja and uplifter fit the bill, and actually our studies match some of what is going on in Jamaica. We now need to look at the other varieties I've talked about, like hockey and Reggie and so forth from Grenada, and those can be compared with the orange flesh varieties, which in many instances have been newly introduced from SIP into the region, and see how they perform in the field. There's been one study, I believe, like that done, comparing pink skin and orange flesh varieties in terms of drought resistance in China, but it hasn't been done in our region as far as I am aware. The other interesting thing coming out of the study was that we couldn't really separate out the pink skin for anthocyanin levels. How much anthocyanin does it have? Is that related to drought stress? Because anthocyanin can also protect you from abiotic stress. But what we do know is that it seemed as if locally, the levels in all sweet potatoes to see what the ranges are. A key point of this study was that it showed us that germplasm conservation is important to maintaining resilient varieties for our region. It also tells us that even though we have different names for these things and we have different morphologies, how we describe them, it's still important to keep those varieties, to maintain them in our germplasm banks, because based on the ravages of climate change in our region, we need to keep these to maintain a good gene pool to produce sweet potatoes for the future. On that note, I would like to thank some persons. First and foremost, Ms. Shannon Gonzalez, who was a research technician on the Molecular Characterization project in Barbados. I would like to thank the CARDI representatives from Dominica, Grenada, St. Lucia, St. Vincent, and Jamaica, all led by Dr. Greg Robin, Dorian, Reginald, Gregory, and Andrea, who supplied our samples. I would also like to thank the PPCR project office at Mona, housed at Maury, Ainsley Henry, Erica Horton, Maxine, who helped with the FedExing of the material and so on, and Jean-Pierre, who was instrumental in setting up today's presentation. So without further ado, I hope that you enjoy the presentation, and I'd like to say thank you for taking the time out of your busy schedule to listen to us today. Thank you.
Okay, thank you so much, Dr. Alleyne. If I'm to speak for the majority or all of us, I would say that your presentation has been very insightful and very clear. We've actually had a request for the presentation to be shared, so I don't think you'll have any issue with the slides being shared.
No, I have no problem.
Okay, and now we'll take some questions. So to ask a question, just raise your hand and wait to be acknowledged. After you're unmuted, then please state your name and ask the question. Okay, so do we have any questions? Okay, oh, all right, so Peter Gay Chang, she says she doesn't have a mic, but she's asking how many varieties are in Jamaica?
I think Dr. Gregory Robin can answer that question because that's some information from CARDI.
All right, so Greg, do you have that offhand?
What was the question?
The question is how many sweet potato varieties are in Jamaica?
Wow, I don't really know. I know the varieties we work with, and the varieties we work with were selected even before I came to Jamaica for specific reasons for research. So the ones we work with are the Ganja, the Uplifter, the Clarendon, Yellow Belly, and one or two others. These are the ones we work with.
Okay. All right.
But there are many varieties, there are many accessions of sweet potato in Jamaica.
Okay. If you don't have a mic, you can also feel free to ask your questions in the chat.
just to add to what was just said, for example, I know that in Barbados, we have about 31 in our germplasm bank. 31 or 32 varies because we may have some duplications. And in St. Lucia, I see they also have about 30 in their germplasm bank. But this may vary based on the fact that sometimes you will have the same name duplicated in the bank.
Okay, all right. Okay, so I'm not seeing any raised hands. So with that in mind, okay. So thank you everyone for joining us. The recording of the webinar will be made available by the end of this week. I'll also include links to both presentations for future reference. So to our funders, the Climate Investment Funds, the Inter-American Development Bank, our project partners, CARDI, our speakers, and the main presenter today, and everyone else who took the time out to attend, I would like to say a very special thank you. I hope this webinar has proved fruitful and insightful to you. Thank you again for your time, and do enjoy the rest of your week.
Thank you very much.
Oh, hold on, we have one question. Okay, let me. Marcia Lord is asking how many varieties are in Grenada.
So I see from a CARDI list 4, but we were given 5. So there may be more than what is stated officially.
Okay, and we have a question from Gerard. Gerard is asking how many, if we know how many varieties are in Haiti.
So Haiti was an interesting situation. When we began looking at this project, we made contact with Haiti and tried to get samples, but they were going through their problems and we couldn't get any. So we don't have any information on how many are available in Haiti. At least I don't. I don't know if Gregory does.
Hey, Gregory?
Yeah, just.
Hold on, Greg. Okay. You may want to repeat that.
Just one comment. Concerning the, I see the participants are asking how many varieties are in the countries. And so, but basically what we did a survey with the ministry, the marketing boards, the farmers, the exporters, and we found out through a very simple survey, which is the most important varieties in the countries. And we selected the top 5 for the study because it's not possible to do and work with all the accessions and varieties in the country. So we selected, from the country's perspective, the 5 most important varieties. And
Okay.
then we worked with those. So it's not a project selection, it's not a CARDI, it's not a UE selection. It's a country selection.
Okay, all right, thanks,
those
Greg.
varieties. Those varieties are supposed to be placed in the germplasm bank, and then later on we can get them to be stored in the International Germplasm Bank at the Potato Center in Peru.
Okay.
That's right.
Okay, all right. So we have a question from Joel Fraser. So Joel, I'm going to unmute you now, and you can ask.
Wait, please. No, listen to this. I understand you. No. Listen to me. How could I come into my... No, how could I...
Okay, hold on. All right, Joel, you may need to unmute yourself. Okay, hold on. Not able to unmute from here. All right, so there's an answer in the chat. They're saying there are 27 lines in the germplasm collection in Jamaica. And there are 2 more questions in the chat. One is, I think, Greg, you may be able to take this one. Is what's the best way to fertilize sweet potatoes? Your mic is muted, Greg.
What's the best way to fertilize sweet potatoes? What we normally do, we fertilize at planting or just after planting when there's root initiation. And then just before tuberization, or just at tuberization, we give another fertilizer application. We do a split application. And one is at planting, just about root initiation and formation, and the other one at tuber initiation. Placement is a few inches, 6 inches away from the vine itself. And we recommend you cover the fertilizer. You put it in a circle and you cover it. In some instances, in large commercial production systems, they fertilize once and they fertilize based on the crop requirement. So based on the crop requirement, the soil and leaf analysis, they will put down a formulation and that would do for the crop.
Okay, all right, thank you, Gregory. So I hope that answers your question, Garth. Okay, still trying to unmute you, Joel. Okay, all right, here we go, Joel. So you can ask your question.
Sorry, that's me?
Yes, that's you.
Okay, so I'm curious about the fact that it is only the white varieties that are drought and salt resistant, because I've always been told that it's the yellow variety flesh that is the most nutritious, and we're talking about making the region more sustainable in terms of food and nutrition. So is that a problem, the fact that it's mainly the white variety? Because I'm assuming they have less nutritional value than the yellow variety. And is there going to be work to try to graft or to put a particular emphasis on trying to get more yellow variety, more of the yellow flesh variety that is drought resistant, because they're supposed to have more nutritional value?
Right. So that's a very good question. And that was interesting for us as well, because we know that many of the orange or yellow type ones, certainly the orange ones based on Gerard, have been recently introduced in the region based on nutrition. But the St. Vincent varieties were pretty interesting because they resembled our traditional varieties, but with yellow skin, and they did not have that drought tolerance hit. So my question is, for me, was really and truly what's happening there. It may involve some breeding trials and some more information on how we can get those genes into the orange or yellow flesh ones, bearing in mind that even the texture and so forth of the white flesh compared to the orange flesh varieties that are high in nutrition is very different. The texture of our traditional varieties tend to be a bit harder and drier with more fiber than the other orange flesh, yellow flesh varieties. So this will require some more study and probably some breeding experiments, and even I would have to say genetic engineering at some point.
I see. Thank you.
Just to add, moderator, just to add to what Angela just said, the Beauregard, the orange flesh, high in nutrition. However, when I was in St. Vincent, we had some plots. It was not an experiment, but we had some germplasm plots. And that's the year we had this prolonged drought. And the Beauregard completely died out. Whereas the other genotypes, which Dr. Alleyne is talking about, survived. And it clearly showed that the orange flesh Beauregard was just not drought tolerant. It would get wiped out in the drought. The vines would just die back.
Okay,
And
all right.
there have been studies done in, like I said, there's one study done doing that same comparison, and they did show specifically that the orange flesh varieties was not drought tolerant.
Okay, all right, so there's a few more questions in the chat before we get to you, Dr. Francis. So the first question is, do you have any information on the use of sweet potato flour to make bread? And if it sticks together like how gluten does for bread flour? I mean, that might be a question for Greg. Greg, your mic is muted again.
Our experiences with sweet potato in making composite bread is grated fresh sweet potato, and we use the Beauregard. It's not flour. And we mix it in a 40 to 60% ratio with wheat flour. And we use a master baker from Trinidad to demonstrate that to bakeries across the region, but it was not flour. So I don't have that experience.
Okay, all right, good. Okay, there was also a thank you from Ministry of Agriculture, Grenada. We thank you for attending as well. All right, so Shanita is asking, for the purpose of the drought tolerance study, how was the drought condition set up?
Okay, interesting question. A typical drought tolerance study would have to be done in a greenhouse where we control rain and we use pots. But in the PPCR project and working in the climate service group at Mona, we're doing a set of studies on the outside. And what we do, we establish the sweet potato crop by giving equal amounts of water for a month. So we establish what we would consider the drought tolerant, the irrigated plots and the non-irrigated plots are all established with water. Once the plots are established, remove the drip lines from the unirrigated plots, and no water is provided after that, except if it rains, of course. So that's how we conduct the drought tolerant trials in the open field.
Okay, all right. Thank you so much. Okay, now we'll get to, all right, so Dr. Marlon Francis, I'm going to unmute you. I'm going to ask you to unmute yourself because the unmute function seems not to be working. And you can ask your question.
Well, first of all, I want to say how pleased I am to see that this particular study is being done. I'm a Barbadian agronomist, and always felt very strongly that these are the kind of studies that needed to be done for sweet potato in particular, because my view is that if there's one plant that will grow on the moon, it would be sweet potato. And I'm fairly confident that when all is said and done, it would be probably the one plant that will grow on the moon. So my question to Dr. Alleyne really has to do with whether or not whether the research has shown any association between the genomic characteristics that have been looked at and the physiology of droughting and the mechanisms of physiology for drought tolerance. In other words, is there a relationship, or has there been a relationship seen between characterization and the physiology of drought tolerance?
Okay. So you're asking if there are any other studies done on that. So
well, if your study have seen, if you've been able to see or throughout the region, if there's any correlation, if anyone's looked at that.
No, not yet. Like I said, this was a simple molecular analysis. We were looking for the genes, and what came up in terms of hits when we did our reference genome matches. We then built family trees and similarity profiles based on that. It is now requiring of us to go and do those studies. Like, for example, what Dr. Robin was saying, you have the drought tolerance conditions, you then extract maybe, you then sequence these two comparative types and do the subtractive analysis for the genes or genomes, see if there are any mutations and that kind of thing. You can do that, but you have to have those controls to do the comparison. We were not looking at that.
Wonderful.
But that's to be done.
All right, thanks for your question, Dr. Fraser. All right, there's another question. Is there a specific reason why Jamaica's productivity is so high? And if it's related to any specific variety?
I think it's simple to adopt the technologies that are transferred by the Ministry Extension. That's simple it is. I think in the islands, sweet potato is still a marginal crop that is not given a high level of management. I think once they put on the management, the marketable yields will improve, because you can get a yield, but the marketable yield, if they have weevil or grub, is going to be significantly affected. So the yields that are quoted are really marketable yields.
Okay.
Normally get that information from export.
Mm-hmm.
All right.
Okay. There is another question from SP Urban. I'm not sure who that is, but it wasn't the uplift variety an exception to the fact that yellow flesh varieties aren't drought tolerant?
Yes, yes, it was. Uplifter was an interesting one. It did show us that it had yellow flesh, and interestingly enough, the trials did show that it had some drought tolerance. So it would require us to do some more investigation as to what the anthocyanin levels are like in Uplifter, for example.
Mm-hmm. Okay, there's another comment from Lisa Myers. So to Dr. Alleyne, although the specific genes for anthocyanins and ABF4 were detected, would we also need to know how these genes are regulated and expressed in the varieties? So she was saying that field trials and further studies may be applicable.
Definitely, definitely, definitely. You want to see how these genes are turned on or turned off. And so this is where we get our comparative studies. So you do your field trials. You also can extract samples along the way and do some more sequencing to do the comparative analysis.
Okay, all right. Thanks, Dr. Alleyne, and thank you, Dr.
Just
Myers.
Angela, you suggested destructive sampling along the crop cycle?
Yes.
All right, because that's going to lead to very large experiments, huh? When you do destructive sampling.
So destructive meaning you sample along the way, not destroying the entire field per se, but you sample along the way.
Right.
I wouldn't call it destructive.
No, I understand, yes. All right.
All right, so Craig Thomas from IACA in Antigua and Barbuda, he said the composite bread making has been executed through capacity building activities with a number of producer groups and government agencies, a number of time in Antigua and Barbuda. And the bread is very well received. It's actually an activity that's conducted annually since the APP project. All right. Okay, so there's a thank you from Shanita to Dr. Robin. She says she has great interest in such studies, and thanks to Dr. Alleyne as well. Looking forward to the continuation of the studies in sweet potato in Dominica. From G. Rowe, excellent presentation and a good example of collaboration. Kelly Brathwaite has a question regarding the C104 Barbados variety. Since the drought tolerance gene was not found, what genetic factors may otherwise have contributed to its growth over the years in Barbados?
So again, Kelly, that's an interesting question. I don't know per se. I believe that in Barbados, you have a mixed sort of cultivation. You do have persons who farm, who grow sweet potatoes, and they do irrigate them on their plantations. So it could be, although Barbados is a water scarce country, that in growing sweet potatoes on these large plantations, as they've done over the years, there's some irrigation given by some farmers. And so that may be one of the reasons why I'm not too clear on that. Again, we only had one sample from Barbados, so I would suggest that I know we have about 30 lines or accessions in our germplasm bank. Barbados itself would require a more distinctive study looking at all their types to see how they tease out with regards to that particular bit of information.
Okay, there's a question also from Victor. If there's any information on Antigua and Barbuda. Greg?
Can you repeat the question?
The question is if there's any information on Antigua and Barbuda.
Unfortunately, Antigua and Barbuda was not included in the study because they're not a pilot country. But I want to say that I have, with the CARDI team, root crop team, we've planned future studies which will include all the countries that were not included in that study. And Antigua has one of the largest accessions, which we've morphologically characterized already. So it's very interesting to see how they match up with the molecular work. So Antigua, be a bit patient, and that you'll get those results one of these days.
Okay, all right. So Lisa Meyer sends thanks, Dr. Alleyne. Okay, thank you everyone for attending. Thank you for your questions, and the slides and the recording of the presentation will be shared, so you have it for future reference. Enjoy the rest of your day and the rest of your week. Oh wait, one more question. Okay, all right, we'll take this one as the last one. Okay, so Gareth is asking, any advice on how to control sweet potato weevil? And if there are any studies done in Trinidad and Tobago?
Well, that technology is known. We use pheromone traps and we use some cultural practices. But I think what's important is where the traps are set. Because sometimes farmers set the traps in the field, in the middle of the field, and that brings in the weevil into the field. And if you have farmers neighboring, you have to do a peripheral trapping. So the traps are placed on the outside of the plots. And they have to be placed on the outside of all the plots to prevent them from getting into the field. So it requires some kind of communal effort if farmers are close by. If you have one single farm in the middle of nowhere, you set your traps on the periphery to keep the weevil from getting into the plots. Okay. But the density of the trapping, the kind of pheromone that's used, all that information is known. If you go to your plant protection unit, the extension unit, you'll get all that information. There are different types of traps. They've been evaluated so you can select which trap you want to use for the conditions you're in.
Okay, all right. So I think that is it for the questions. If you have any further questions, just send them to the email that would have sent you the invitation. We'll put them all together and send out a single email with all the answers. Okay, thank you.
Very much.
Okay, thank you, moderator.